So it's my pleasure to introduce our keynote speakers for this afternoon, Dr. Michele Myers and Dr. Pinar Cicalese, both from GlaxoSmithKline. Dr. Myers is the Senior Director of Cell Process Development for Cell and Gene Therapies at GSK. She's been with the company a long time and has been instrumental for the company gaining approval for a novel gene therapy treatment called Strimvelis, which they will talk about today. Dr. Pinar Cicalese is the supply chain manager who develops the supply chain strategies for the cell and gene therapy program at GlaxoSmithKline. I was remiss in mentioning Michele's qualifications, so I'm just going to say a little bit about your background, if you don't mind. So Michele received a BA in biology from Temple University and has a PhD in biochemistry from Penn State University. Dr. Cicalese obtained her PhD in chemical engineering from Drexel University, and has a master's in science and a bachelor in science in chemical engineering from Bogazici University in Turkey. So it's my pleasure today. Both of these ladies have been great partners with the biotechnology program here at the college, and I appreciate them always being willing to come out and talk about what's going on at GlaxoSmithKline. So thank you.
Right, everybody can hear me? Good. Today, Michele and I are going to talk about advanced therapy medicinal products. These are the products that are different than what you are used to. They are not small molecules or even biopharms, like they are not monoclonal antibodies. We are going to talk about what those are and how GSK is involved with these advanced therapy medicinal products, especially in cell and gene therapies. And we are going to go into how do these cell therapy modes work and how they are manufactured, career options, and finally we'll wrap it up with a key takeaway. So traditionally you are used to seeing small molecules like tablets or pills and proteins even have been almost a household name in the past 20, 30 years. However, advanced therapy medicinal products are just coming up. I'm going to go into the field a little bit. They are either cells that are manipulated to express the gene of interest or a gene of interest DNA that is delivered to the patients via a vector. So I really like this slide because it brings home to me. In the top, you can see some of the medicinal products: aspirin, somatropin, and Herceptin. Aspirin, everybody knows, is a small molecule. Somatropin is a human genome hormone, so it's also familiar to some people. And Herceptin is trastuzumab, which is used in breast cancer. So it's a monoclonal antibody. And comparable objects from our daily lives are a bike to a car to a business jet. And you can see advanced therapy medicinal products are even further advanced than those, and therefore it's the complexity, relative cost of development, the amount of complexity to deliver this to the patients increases. This is some statistics from a consortium called ARM, Alliance for Regenerative Medicine. They are mainly the biggest consortium in this area, advanced therapy medicinal products. And in the top, you see the 2018 total global financings. And if you look at gene and gene-modified cell therapy products, year-on-year increase, so from 2017 to 2018, there has been 64% increase in the financings of these products. It's in the billions that companies raise money for developing and bringing these products to the market. And you can also see on the bottom right-hand side, it is how many clinical products are going through, clinical studies are going through right now. And you can see the phase 1, phase 2, phase 3 breakdown. And most of those clinical studies are in gene therapy and gene-modified cell therapy area. Are there products in the market? Yes, there are a handful of products in the market. Strimvelis was one of the first products that was approved in Europe, and it was developed by GSK and brought to the market by GSK, and it was for a severe immunodeficiency, bubble boy syndrome, and it was approved in 2016. Afterwards, GSK divested this to Orchard Therapeutics. Kymriah and Yescarta are the other ones that you may hear. Sometimes news about these come up in the scientific community, and those are also approved in 2017 and 2018, actually. So approval of these products are ongoing, and as you see from the clinical trials, there will be more and more approvals in the upcoming years. GSK's initial involvement in cell and gene therapy was with rare diseases, and as I mentioned, the ADA-SCID, the severe immunodeficiency, the bubble boy syndrome product was approved in 2016. And there is also other products that as a whole, as rare disease unit was divested by GSK to Orchard Therapeutics to be able to focus on oncology products. And that's the future currently in cell and gene therapy, of cell and gene therapy in GSK. So what this means is any type of cancer that you may be able to treat with these type of gene or gene-modified cell therapies. And what you do is, in the next slide, autologous or allogeneically, you will be able to modify the cells. And what does it mean? Autologous is what we are focusing right now. And these type of therapies take immune cells from the patient, either as a whole blood or immune cells, and take it to a manufacturing site and modify these cells with the gene of interest, and then take it back, give it back to the same patient. So you can see it's a personalized medicine, therefore there is less risk of immunogenicity because you are treating the patient with their own cells. However, there's challenges in manufacturing because one patient is one batch. So if you can imagine, there shouldn't be any mix-up because if you give other people's cells to a patient, that's obviously a big risk. Allogeneic, on the other hand, has what it means is there's one donor, you take the cells from that donor and modify it, as you see, as needed for the disease area, and then you deliver it to all the patients. So there's only one donor that goes to all the patients. Obviously, this type of manufacturing is easier because you only have to start from one starting material, and one batch can treat multiple patients. However, the immunogenicity risk is higher in this case. So there is real challenges, as I mentioned. If you can think about one patient, one batch, this means you can't really scale up there is really no economies of scale like in biofarms. However, there is economies of scale in scaling out, meaning rather than operating one room, one patient, one batch manufacturing, you may be able to operate multiple equipment in one area, and again, be able to treat patients from one manufacturing site in that manner. So real-time tracking is a big challenge, obviously, because from the time the cells leave the patient's location to the time it comes back to the patient, you have to have tracking and you have to have control of these cells. Logistics is a big challenge, and transportation, because if you imagine all the snowstorms we had this year, if there's a snowstorm and you have to transport patient cells, you're stuck, right? So you have to put your manufacturing centers in a certain area, imagining all these type of logistical risks as well. And packaging for primary and secondary needs to ensure product integrity as well. As the patient's numbers increases, you can see how these challenges multiply. Normally, for rare diseases, patient numbers can be a handful per year. And if you go into oncology, which most of the cell therapy products right now are in liquid type of cancers. So-called liquid is blood-based cancers, where the reaching to the tumor is easier. However, we are also working on treating solid tumors. So you can imagine, once we have proof of concept, how many more diseases we can treat with these type of therapies, and the patient numbers obviously multiply to the thousands and maybe to hundreds of thousands. With that.
Thanks. Okay, thanks, Pinar. So Maggie asked us to incorporate a few slides to talk about the technicalities of how this all works. So I've got a few slides to try to walk through this. Hopefully it'll be a story that you can take back as you're talking to students about some of these basic things, that you can say, oh, this is how it works, and this is what it means, and this is what folks are doing with this technology. It starts with the central dogma of molecular biology, right? That DNA is transcribed to RNA, which is translated to protein. So we start with that basic premise. And as Pinar was saying, for example, with Strimvelis, for ADA SCID, the severe combined immunodeficiency, what was discovered was that severe combined immunodeficiency was the result of missing one protein in these kids' cells, which compromised their immune systems and that created this bubble boy syndrome for them. So the thinking was, let's use our molecular biology tools and, oh, sorry, not where I wanted to go, and fix the DNA, right? If you can fix the DNA and put those corrected cells back into these kids, you can cure their severe combined immunodeficiency. And we got Strimvelis approved because it worked. Lo and behold, when you fix the genetic code, it worked. And there were 18 patients treated before it was divested from GSK, and I think don't quote me exactly on the number, but at least 16 of those kids were in school. Those kids were cured and were able to go to school. Knock wood, I've not heard of any follow-up, again, because GSK has divested those programs. But the cure rate with this therapy was just fantastic. And regulators, the folks in the FDA, the folks in the European Medicine Agency, were all over it when GSK took responsibility for this because they wanted it to get to the market. It was needed. These kids were really dying, and so then there was no other real therapy for them. So it was all about fixing the DNA, and now the thinking is fix the DNA and incorporate into that DNA molecular or a target, or what's the word I'm looking for? Incorporate into that DNA a protein that will recognize tumor cells. So that's the way that Yescarta and Chimera are working. Incorporate into the DNA something that will recognize a tumor cell and kill it. And you know what? It works. For 85% of those patients who've been treated with Chimera, it works. That's ALL is the primary indication, and you've seen these headlines in newspapers and stuff. It works. So there's a lot of enthusiasm in this space, and as Pinar was saying, there's a lot of money pouring into this space. So that's why companies like GSK and other small biotech companies are really investing here, because you go back to the central dogma of why it should work, and yeah, it really should work. So how's that, what do you mean, just fix the DNA, right? Is there really a pair of tweezers? And the answer is no, of course it's not quite that easy. The answer is HIV. So there was a Dilbert cartoon once I saw where it showed this guy, we're going to cure your cancer with HIV. And you're like, you know, your hair pops out. Well, what are you talking about? It turns out that viruses are pretty simple organisms. I'm not even sure they count as organisms, but they, right? They're pretty simple. And the fact of the matter is they're only here to reproduce themselves. And to do it, they don't need much in terms of their genetic code to reproduce themselves. There's just a few proteins, just a few things in their genetic code for a few proteins that they need to reproduce themselves. They need to make their outer coat, they need to make some enzymes, and they need to reproduce that nucleic acid. And that's really what they're all about. So with these molecular biology tools that are out there, someone got the brilliant idea, and I certainly don't take credit for it. There are very smart people all over the world who've done it. But for example, Strimvelis was first recognized in Italy, and those researchers identified how to change this virus to do what they wanted it to do. But the virus itself, again, pretty simple biology. And I don't claim to be a virologist. This is as simple as it gets for me. The virus attaches to a cell, it goes into that cell, and it takes its genetic code and it takes over the nucleus, right? It takes it over and inserts its DNA into the DNA of that host. And then it takes over the factory, and it just starts making more of its proteins, more of what it needs to make in order to reproduce itself and make more virus, right? So the idea is take control of this. Use this virus for good. And so if you took the code out for those proteins that it needs to reproduce itself, and instead incorporate the gene that codes for the protein that is missing in these kids with ADA SCID, right? Encode for that gene and take the part that make it infectious. Take those elements that make that virus infectious out and incorporate the gene that you want, and then give it those genes separately. So in a separate system, what we've done is incorporate these elements that the virus needs to make more, and use this mechanism, use the what's normally in the virus to make the gene that we want to make. And then what we end up with is a virus that just has the gene of interest in it, right? It's just got that gene of interest. It doesn't have all of the other machinery to make itself infectious, right? So when you've got this virus, it's not even a virus now, it's a vector, right? You've got a viral vector that's got our protein of interest in it. And take it and now take the cells from these kids, right? Identify their stem cells, pull those stem cells out, and have this viral vector transduce it with the gene that they're missing. So the virus attaches to the cell like it normally would. It puts in the genetic code that we want it to put in now. And now these cells are going to go through and use the machinery from DNA to RNA to protein to make the protein that's missing, but because it doesn't have any of those other elements to make it infectious, it's not going to make an infectious virus, right? So now that cell is chugging out the protein that it was missing. And if you incorporate it into the stem cells of these kids, then those stem cells can go on and create other cells and have that sustainability within the body. And that's what's working in these children. Yeah? So, and the same concept is true for cancer, for oncology. So take the cell, incorporate into it what we want it to express. We want it to express a chimeric antigen receptor. You've seen this terminal, a CAR. CAR T cells are the big thing, capital C-A-R. CAR T cells are the next big thing. It's a new medicinal form, right? You have small molecules, as Pinar was saying, the small molecules, the proteins. Now we have these CARs, chimeric antigen receptors. So this chimeric antigen receptor or some other receptor that we can engineer onto the cell is incorporated into a patient's immune system, into their T cells. Take the T cells from the patient, incorporate this. Now, the virus doesn't have what it needs to be infectious, but it has what it needs to make this receptor that is going to target the tumor cells within the patient. So this is the technology. Hopefully, if you took this conversation back to a student, you could get them excited about, well, this is why it's important. The DNA to RNA to protein. You can make that a story that cures cancer. Right? Again, what happens is that change in the DNA is in that virus. The cells come from the patient. We treat the cells with that modified viral vector, and that stays in those cells. Those gene-corrected cells go into the cells that are isolated. They go back into the patient, and years later what they found is that the recovered gene continues to be present. That's what they found in the children who've been treated with Strimvelis. So I think it's really remarkable technology. It's exciting for those of us working in this field. It is, as I said, a very exciting time to be here. There's great opportunity in this area. Of course, with great opportunity comes great expectation to deliver and get these products out to the market, get them in the clinic, and demonstrate their efficacy. Again, how do we make them? First, we've got to make that viral vector. As I was explaining it to you, what's done is it's a process very similar to how proteins are made, where we have a cell that is transfected. So we add in all of the genes of interest in a transfection, and then we harvest that viral vector, purify it, and freeze it away until we're ready to take the cells from the patient and use it with the cells from the patient. And then we have a separate process for using those cells from the patient, where we isolate the cells, we treat them with that viral vector, and then we expand them. So, of course, there's challenges in all of this. We're creating a process to do this, but the incoming material is always going to be a little bit different. And so we've got to find a way to make that, to manage that challenge of having that input material always a little bit different. So that's why it's taking time. Actually, while our clinical colleagues are eager to get these therapies into the clinic and to demonstrate their efficacy and to move things along, those of us who work on the production side are a little more hesitant because we want to make sure we're making it the right way to make a safe and efficacious product. So, how do we do this? How do we go from this brilliant idea that you can change the DNA to RNA to protein and turn it into a medicine? And it takes a number of years. If you've seen this in the literature, it takes a number of years to go from research and development to manufacturing and to bring all of the analytics along with it. So before we close out the presentation, I want to just kind of walk through what that looks like within GSK. Starting with research and development, where essentially it's discovery. Identify the potential medicine and its disease target and its mechanism of action. What do you want to treat? How do you want to do it? Generate data to demonstrate that it'll work, and then figure out how to make the drug. So this is all happening very early in the process. Who do we need to do this? So bringing it home to how to get students enthusiastic about this and what kinds of jobs are out there for your students. Immunologists, biologists, biochemists, microbiologists, just about any science field you can think of. Bachelor's degrees, master's degrees, pharmacists, and certainly those with graduate's degrees, especially in early development, the research and discovery arm. It's really those folks with undergraduate degrees, but a bachelor's degree is really sufficient to get started in any of these areas to start contributing to the discovery efforts. Once we get through and we've ticked these boxes, we need to start a clinical trial. Well, what do you need to do to start a clinical trial? You need to convince the FDA that there's a reason to believe. So you've got to amass that data. What do you need to do that? Well, you need everybody who's been participating in research and development, plus you need regulatory affairs, you need quality assurance, you need physicians, you need pharmacists. What's it take to be in regulatory affairs, quality assurance? So these are additional jobs that are out there for folks who have science background. If you have a science background, then you can understand what's important to control to ensure the quality assurance of the product, to manage the conversations with regulatory. Those are all jobs that need to be filled in order to bring these medicines to people. Once you're in a clinical trial, you've got to manage that trial, you've got to make the medicine, so you need all of these people, plus you need production operators, you need quality control, the people who are testing it before you can actually release it to a patient, clinical trial managers, physicians. So here, when we start to talk about production operators, quality control analysts, I think those are the people that are coming out of programs like Maggie's here at Montgomery County Community College. You can do that with an associate's degree. We have folks who are working in production as production operators, as analysts, with undergraduate degrees and associate degrees. Those jobs are out there for those who are excited about this technology. Once we get through the clinical trial, we've got to analyze the data, so we've got to evaluate that clinical trial data, and we've got to demonstrate that we know what we're doing in production. We've got to demonstrate that we can make this medicine routinely in a safe and efficacious way. What do we need to do that? Who needs to be involved? We need all those people from R&D and quality assurance, statisticians, physicians, production operators, the quality control analysts, validation engineers, process engineers. There's a job for everyone. It takes a village to raise a kid. It takes a big village to make a medicine. Once you do that, you've got to apply for a license. You've got to convince the regulators that your medicine is safe and efficacious. You need the clinical data to do that, and you need the production data to do that. So you need everybody who's been involved to date. Regulatory affairs, quality assurance, the manufacturing personnel all have a role to play. And then once you get it approved, you make your medicinal product manufacture. And, of course, you can't just keep it the way it is. You've got to manage it throughout its life cycle. You've got to look for ways to improve its production. You've got to look for ways to minimize the costs associated with that production. So there's all of that work that has to continually happen. So you need all of those people that we've talked about before, plus you need environmental monitoring to go on. You need calibration and maintenance of all the equipment that's used. You need technical support to make sure that anything that goes wrong is investigated properly. Supply chain, procurement, IT and automation, data management. We are generating reams and reams of data. Getting our arms around that data is a huge job. Ensuring the integrity of that data is a huge job. A facilities engineer. So my point in sharing all this is to drive home the message, hopefully, that this technology is exciting. It's based on principles that you talk to your students about, and there are jobs out there if they're excited about this technology that they can get involved in. So the key takeaway is science has evolved to enable gene therapy. Cell and gene therapies hold great promise, especially for things that have otherwise unmet medical need. And companies need people. They need trained and motivated people to contribute to making these ideas reality and making these medicines happen. So we'll close there, and Pinar and I are happy to take any questions. Yeah?